The plasma membrane acts as a barrier that protects the cell and helps maintain the correct concentration of solutes in the cytosol. And likewise, the membranes around the various organelles help protect their internal environments. Biological membranes consist of a layer of lipids and protein molecules, with roughly 50% of each. In the membranes, the proteins are larger than the lipids, with about 1 protein molecule per 50 lipid molecules. In the membrane, the lipids provide the barrier, and the proteins, which may span the membrane or may be attached or may sit in the membrane, provide the function. Examples of what the proteins can do are transporting ions or molecules, catalyzing reactions, having structural links in supporting the shape of the cell or the membrane, or they may act as receptors to convey information from the outside of the cell to the inside. Basically, the proteins add functionality to the membrane. They allowed inside things to get out and outside things to get in. And as the cell can control the level of the proteins in the membrane, it can effectively therefore control the flow of material and information. However, before we get to that, we need to look at how the proteins interact with the membrane. In terms of the interaction of the proteins with the membranes, we have 8 formats. 1. Single membrane spanning proteins. These proteins can have either the N or the C terminal outside the cell and have an alpha helix that spans the membrane. An example of such a protein is a receptor that binds a ligand, a message outside the cell, and then transmits a message to the inside of the cell. 2. Multiple membrane spanning proteins. These proteins have multiple alpha helices across the membrane. An example of this type of protein is the G protein coupled receptors, the GPCRs. They have 7 transmembrane spanning domains. 3. Rolled up beta sheet proteins, also called beta barrels. In this format, the protein forms a barrel or pore using beta sheets that span the membrane. An example of this type of structure are the regulated pore or channel proteins that allow material in or out of the cell. 4. Embedded alpha helix. Here, the protein is anchored to the cytosolic face of the membrane using an embedded alpha helix. An example of this type of proteins are the reticulins that are involved in the shaping, that is, the curving of the membrane of the endoplasmic reticulum. 5. Covalently bound lipid chain. In this case, the protein has a covalently bound lipid molecule. That is, the protein is mysterylated, palmitoylated or prenylated, and the lipid chain, being hydrophobic, forms an anchor in the cytoplasmic face of the membrane. Examples of this can be found in the alpha and gamma subunits of the heterotrimeric G proteins, which are involved in signal transduction across the membrane. 6. GPI or glycosyl phosphatidylinositol anchored proteins. These proteins are found on the outside of the cell, and the protein is anchored to the membrane via the GPI link. Examples of this type of protein can be found on the surface of oocytes, where they bind to their counterparts on the sperm. 7. Cytosolic membrane-associated proteins. These proteins are attached to the membrane via another protein. An example of this is a cytosolic insulin receptor substrate that attaches to the membrane-spanning insulin receptor, and the membrane-spanning insulin receptor is another example of the class of proteins I first mentioned. That is, single membrane spanning proteins. And finally, 8. External membrane-associated proteins. In this case, the protein attaches to another protein that spans the membrane. An example of this type of protein is the toll-like receptor that binds bacteria proteins as part of the immune system. Of the protein types I have just mentioned, 1, 2, 3, 4, 5 and 6 are referred to as integral membrane proteins and can only be separated from the membrane with detergents and some solvents. Types 7 and 8 are referred to as peripheral membrane proteins and can be separated from the membrane by changes in pH or salt concentration, or just by giving the sample a really good hard shake. Some of the membrane spanning proteins may provide anchor or docking points for other proteins, and I've already given you an example of this with the insulin receptor and the insulin receptor substrate. This type of protein will interact with the other proteins by using domains such as the PTB domain, the phosphotyrosine binding domain, and the SH2 and SH3 domains, the Sark homology domains. There is also a domain called the pH plextron homology domain that allows proteins in the cytosol to bind to the plasma membrane. One feature that a lot of proteins have that are exposed on the external face of the plasma membrane is the addition of sugar molecules. That is, the proteins are glycosylated, a process that starts in the endoplasmic reticulum and is finished in the Golgi. And it's not just proteins that are glycosylated. Some of the lipids can also have bound sugar to form glycolipids. This apparent cell coating of sugar residues is called the Glycocalyx, and the sugar residues are involved in cell adhesion and also help protect the exposed proteins.